Thank you so much for having me. It's a pleasure to be here. And hopefully we'll kind of, the answer, long story short, is we still don't know, but hopefully yes. I'll try to convince you whether we have enough evidence that it might suggest that in fact there might be something cooking under the hood. Right, my name is David. I just completed my PhD in the Department of Medicine, based in the hospital campus here in Cambridge. And I was fortunate enough to get one of these wonderful awards on my PhD work. So as a brief introduction, Parkinson's disease is a neurodegenerative disease, and I'll tell you what that means in a second, but it affects over 9 million people globally. It's the second most common after Alzheimer's, which is very famous, and it affects people all over the world. It is a neurodegenerative disease, as I said again. It basically is a disease that affects neurons and that leads to the degeneration or the death of neurons. That's irreversible, as far as we know now. Basically, on the left-hand side, you can see that there's this section of the brain called the midbrain, which is deep inside the human brain. And there's these groups of neurons called dopaminergic neurons that live in this kind of wing shape. Let me see, control. Okay. They live in this kind of wing shape like structure in the midbrain. And they've got this dark coloration, so if you ever see a section of the midbrain, they've got a kind of dark pigment that's natural to them. And in a patient in a kind of normal patient or normal brain, these neurons are healthy and can do their job. But in a Parkinson's brain, these neurons will accumulate these things called Lewy bodies, which are these misfolded proteins that we still don't really understand. We understand a little bit about how they form, but not what's causing them and whether they're beneficial for the cell or whether they're killing the cell. What we do know is that these cells die, and at about 50 to 70% of these cells will die, and that's when patients will show lots of the common motor and non-motor symptoms. Sleep disturbances, tremors, difficulty moving, difficulty with their gait, but also some non-motor symptoms that are less famous, like cognitive deficits, sleep disturbances, and even GI issues that precede the disease even 10 years plus. And unfortunately, there are no curative therapies for Parkinson's at the moment. There's a few treatments here or there that are addressing kind of the tremors, and they're addressing the symptoms, but they don't slow the neurodegeneration. And so for this, it's really, really important to understand the disease mechanisms better and to be able to use new drugs, develop new drugs, or use and recycle drugs that we already have on market. And there's a new kind of perspective to this that my PhD was really centered around. And this is this tissue called the meninges. If you've never heard about the meninges, which you probably haven't, I'm showing you here a cross-section of a human brain. So you can see here on the right. And here we have the brain in the inside, and we have the skull and the skin on the outside. But between the brain and the skull, we have these 3 layers of connective tissue called the meninges collectively, and there's 3 of them, the dura, the arachnoid, and the pia. The names are kind of irrelevant, but basically these 3 layers of connective tissue both protect the brain physically and contain this brain water called cerebrospinal fluid. Basically will prevent your brain from sloshing around in your skull and help prevent injury. But there's a really important function for the meninges that people don't really talk about very often, and this is one for waste collection. And so all the waste of your typical organs, like your skin, your kidney, will go into the blood and using these vessels called lymphatics, but the brain doesn't have those. And for years, people didn't understand how it worked, but basically the brain throws its waste into the kind of brain juice that I mentioned, and that ends up in the meninges. And the meninges actually is filled with immune cells, and people only discovered this about 2015, and have been characterizing it ever since. And immune cells typically, like here we have a T cell in green and this purple cell. They respond in lots of infections. So, in COVID, you'll hear about immune cells, and what happens is things like bacteria, viruses will be eaten by immune cells presented to these T cells in green. These guys will then multiply and express lots of pro-inflammatory factors that will clear the bacterial or viral infection, and that leads to health. But we have another side of the immune system which results in autoimmunity, where you have things that are normally healthy in your body, so self proteins that will be presented to the T cells as if it was a bacteria or a virus, and that leads to disease. And we think that's what's happening in Parkinson's. And so here I'm showing you a diagram where we have the brain at the bottom and the meninges at the top. And so our hypothesis at the beginning of my PhD was that neurons that are dying in the brain due to Parkinson's, and why that happens we still don't understand. We think that that was releasing some sort of factor, whether it was a misfolded protein like I mentioned or something else, we weren't sure. But all of that we knew was going to end up in the meninges, but just by how the organs function. And all the immune cells that are localized in the meninges can kind of capture them with their receptors and eat them. That would then lead to an immune response of some sort, and that would kind of translate into the brain and result in neurotoxicity and cell death. And we think that that was going to propagate the disease. So if that was too much, just think of this as there's a signal coming from the brain into the meninges, and there's a response, an immune response, from the meninges into the brain that's affecting these neurons. And so my PhD started with a few observations. First of all, the observation that lots of these T cells, like I mentioned, that are immune cells, were localized to the substantia nigra in the mouse, shown here. And so these mice, their substantia nigra neurons, dopaminergic neurons, are here in green, and so they have that lovely wing shape that we can see by microscopy. And we see that these T cells often are in this region and even having cell-to-cell contact with these neurons, which we knew was not beneficial. But when I looked at the meninges, and what I'm showing you here, so that's the shape of that in the mouse. We can, if you can imagine the dissection, you remove the skull cap in a dish, and then you peel the meninges off, and then you can flatten it on a microscope slide, and then you can image it very, very nicely. And I'm only showing you the central blood vessel that runs through the meninges. And we stained it for T cells and B cells, which are two varieties of immune cells. And we can see in the control there's very little, but in the Parkinson's model, there's these big clusters of T cells and B cells, which we knew was kind of an indication of an immune response. And in kind of homeostasis in a normal mouse, these can sometimes appear, but they're generally associated with old age or with an infection, and hadn't been seen before with neurodegeneration. We then use this technique. I mean, you can't see it, but there's some statistics there for you. We counted all the cells in the brain and in the meninges with this technique called flow cytometry, and we saw that there was a significantly increased number of T cells in both of these, and that kind of the number of them were almost mirrored, and so we thought that these two organs were really intimately connected. More imaging of the meninges showed that. So here we got the T cells in red. And as I showed you before, in the case of a bacterial infection, these multiply and will then kind of activate. And that will lead to either clearance of the bacteria, but in autoimmunity it will lead to disease. And here we can see that we have all these little foci of kind of proliferating or multiplying cells. And we also see the presence of them kind of embedded with these cells that allow them to activate. And these are called antigen-presenting cells. We didn't know whether they were, because there's lots of flavors of these immune cells. They can be beneficial, they can be pro-inflammatory, which are not great in health. And so we asked whether these were inflammatory cells. And so again, we dissected the meninges off of Parkinson's mice and control mice. We stimulated them in a dish, and then we can read whether they produce these proteins called cytokines, which are pro-inflammatory factors that are here in the X and the Y axis. So in our control mice, these don't express any of that, but in our Parkinson's mice, they do. And when we count them with flow cytometry, we saw that there was a significantly increased number, almost doubling and tripling in some cases, of the inflammatory cells. And so we think that in the meninges, these cells are multiplying, and then they're then going into the brain and secreting this pro-inflammatory factor. And ongoing work in the lab is trying to find out how does this interferon gamma, this pro-inflammatory factor secreted by the T cells, how does it affect all the other cells in the brain? Because there's very, very few immune cells in the brain, but all the other cells like neurons, glial cells, even blood vessels and so on, those will contribute to disease in a way that we still don't really understand. And so we want to know what's the effect of that on the rest of the brain, and how does that lead or contribute to Parkinson's disease. And we already have some clues from late work on my PhD, so I won't bore you with the details of this, but basically we can do this fancy technique called sequencing, where we can untangle all the states of the cells. So if the cells in the brain are sensing pro-inflammatory cues, or if they're normal, and we can see that they're sensing lots of pro-inflammatory cues from interferon gamma, as you can see here, in the Parkinson's mice. We know that these neurons are dying, and we can see that other cells are activated like astrocytes that shouldn't be. So in brief, my PhD basically found that dying neurons in the brain in this Parkinson's model release some sort of factor that is captured by immune cells. It leads to the proliferation of T cells, which are these pro-inflammatory cells, which then can migrate into the brain and can cause neurotoxicity and cell death via this axis. And really importantly, kind of being that it's CSAR, what is the application of the research, and what impact will it have? And so, as I said, kind of in the title of my talk, at the end of kind of these results, we're really interested with my supervisor in creating a clinical trial in Adam Brooks Hospital, as one of the kind of big Parkinson's leaders is already based there. And so we would propose that we would take a cohort of these patients, and instead of treating them with the current drugs that they receive, or in addition to, we would use things like TNF inhibitors, monoclonal antibodies, and drugs like tacrolimus, which are inhibiting either the pro-inflammatory factors or the cells themselves. And so in this kind of diagram, these would inhibit kind of this cycle and kind of this bit of the mechanism as well. And so this would basically result in a novel treatment or a novel way of treating some patients, potentially not all patients, as there'll be some differences between the cohorts. But we'll have a novel way of treating some of the patients who have Parkinson's, who have no other kind of clinical options. And then this would, hopefully result in helping some patients and their families with this devastating disease. And just one final thank you. So there's a kind of cash award that comes with CSAR, and I used it at the end of my PhD to return to Mexico, which is where I'm from, to do lots of science outreach, to recruit students to come to do a PhD in Cambridge. So this is me giving a talk, and I managed to publish some of those kind of strategies that I developed in Nature magazine, which was fantastic, and a kind of wonderful end of my PhD. With that, I'd just like to acknowledge lots of wonderful people that made this all happen and helped with the experiments, and would be glad to take any questions. Thank you.
Thank you. That was a wonderful talk. We've got time for just a couple of questions. Can I slip one in quickly? I do remember, 10, 15 years ago that it was amyloidoses were going to be the root cause of everything, yes, including Parkinson's, but that didn't really feature in your wonderful overview.
Well, normally, sorry, I should probably speak up. Normally, the issues why kind of Parkinson's and all the other neurodegenerative diseases have maybe stalled a little bit with their progress and bring things to clinic like drugs and so on, was because all those people were neuroscientists. And I think they were studying they were trying to understand why genetically and kind of exogenous factors, what is contributing to the misfolding of the proteins, which proteins are misfolding, how can we stop that? Is this beneficial to the cell? Is this killing the cell? And I think immunologists are really good in the context of cancer at identifying kind of big changes that are happening, and then kind of using new drugs to attack that.
It was great, thank you. That was very helpful indeed.
Fantastic talk, David. Thank you very much. And I'm really pleased to see you spreading the word with your outreach in Mexico. That's really excellent. Setting up the clinical trial, that's quite tricky, isn't it, I presume? I presume there are ethical hurdles that you have to leap to get that set up.
Yes, and I can't speak to the logistics of that because I'm not a clinician. My boss is a clinician and she works at Edinburgh. She's an honorary consultant. And we collaborate with a lab that is used to setting up Parkinson's trials. So we were thinking of jumping on his platform to then basically leverage his ethics and his expertise in that. So at the moment, I can't really elaborate, but yes, certainly there's a lot of ethics involved in that.
Well, good luck with it.
Thank you.
Okay, just a question back. Yeah.
I'm interested to hear you talking about Lewy bodies because I've only ever heard of those with regard to one of the forms of dementia before. Is there a crossover then between the Parkinsonian side and the dementia side?
Yes, so Lewy bodies were identified. In fact, the woman who generated the mice that I used for my PhD was the woman who identified the protein in Lewy bodies, and so basically all of those are kind of united within, a big umbrella of synucleopathies, and we still don't really understand how the Lewy bodies are actually contributing to dementia. We know that they do, and whether that is guiding the immune response, we're still not sure.
Great. I think we should
Okay,
stop
perfect.
there. That was a brilliant talk. Thanks so
Thank
much.
you very
And,
much.
again, congratulations on using your money so creatively.
Thank you.